Have you ever walked past a bakery and instantly recognized the sweet scent of fresh bread, or caught a whiff of rain on dry earth after a summer storm? It is amazing how our brains can distinguish between thousands of distinct smells, each triggering a unique memory or emotion. But how exactly does this happen? Well, the science behind different smelling molecules is all about their unique shapes and electrical properties. Every scent we perceive comes from airborne molecules, which are tiny chemical compounds that fit into specific receptors inside our noses, much like a key fitting into a lock. The distinct combination of activated receptors then sends a unique signal to our brain, allowing us to identify and interpret a particular smell. This intricate process is known as olfaction, and it relies heavily on the principle of shape selectivity. Imagine each odor molecule as having a particular three-dimensional structure. When we inhale, these molecules travel up into our nasal cavity and bind to specialized proteins called odorant receptors, located on the surface of olfactory receptor neurons. There are hundreds of different types of these receptors, each designed to recognize and bind to molecules with specific shapes and sizes. For example, a molecule with a complex ring structure might bind to one set of receptors, while a smaller, linear molecule might activate another set. The slight electrical charges on the molecules also play a crucial role, influencing how strongly they interact with the receptors and contributing to the overall scent profile. It is the combination of shape, size, and electrical characteristics that determines which receptors are activated and how strongly, creating a unique molecular fingerprint for each smell. So, to better understand the smells around you, consider the diverse chemical structures. Think about how a complex floral fragrance might contain hundreds of different molecules, each contributing a subtle note by activating various receptor combinations. When you experience a particularly strong or subtle smell, try to mentally picture the tiny, invisible particles that are interacting with your nose. You can even experiment with common household items like different spices or fruits, noting how their distinct aromas relate to their underlying chemical makeup. This molecular recognition system is incredibly sophisticated. Researchers use a technique called gas chromatography mass spectrometry, often referred to as GCMS, to separate and identify individual odor molecules from complex mixtures. This allows them to precisely map the chemical components responsible for a particular aroma. Furthermore, the concept of stereoisomer, which are molecules with the exact same chemical formula but different three-dimensional arrangements, can lead to drastically different smells. For instance, one form of the molecule carvone smells like spearmint, while its mirror image smells like caraway, showcasing the incredible precision of our olfactory system. Ultimately, recognizing that every scent originates from specific molecular interactions empowers you to appreciate the hidden chemical world shaping your daily experiences. It is a constant dance of microscopic keys fitting into microscopic locks, translating the invisible into the vivid tapestry of aromas that enrich our lives. Understanding this fundamental process truly reveals the amazing complexity and precision of one of our most fascinating senses.